Does what look okay? I have a proposal.
Well, what is it for Nora?
You just like shred them.
You were so upset. I thought so.
Well, if you're gonna give us points.
I know you're going to earn points. I have picked out 28 of the worst questions. That's right.
Okay.
That I am going to let you. If you can get them all right, you'll get 14 points.
All of them.
So cap.
I'll give you a hug too. Well, I'll give you a hug.
I'm not touchy feely.
Okay.
Everybody knows that.
What about
I am
air
like,
hug?
I
Are you
am-
like a candy person?
I'm like not. Everybody, you meet somebody the first time and they hug each other.
Ugh.
So I'm not a German.
I am.
But I'm just not touchy feely. So, said my wife. She's
Do you like to hug the dog?
yes.
I will hug you.
See, every cat for 11. So we have 28 questions here. You have a scan chart. Get these done. I'd like to have them by Wednesday.
Come on, guys. Wednesday.
So there should be 2, 5, 6, 8, 11, 14, and 19. So everybody here is taking the test, right? Because I think everybody.
Yeah.
I have one student who has not.
Uh,
Right. And Russ had a very good reason, unfortunately.
Yeah.
Very sad. Very sad. So please
We love
don't. And oh, guy's wonderful, but that's extraordinarily sad. So just make sure you don't show it to him. I appreciate that, please. And get those. So just give me back the Scantrons with your name on it, and so I know who's who. And I have for both classes.
I'm very happy.
Here's my comment: study harder. And the other comment is, we have to look at, and maybe we'll take a couple of these questions after this is done, maybe by Friday, and say, why are we getting these wrong? Okay, why? Because to me, a lot of it is less. I was saying to a colleague of mine this morning, why I don't accuse people of not studying. And I certainly would, because that's not correct. I have no idea. I mean, everybody knows what they did to prepare. But the reality is, I see a lot of problems with two things. Not understanding good test taking strategies and question analysis, which quote unquote means reading the question properly, and some other little odds and ends. And the other one is test taking anxiety, which is rampant for some reason. And because I realize there's a lot of pressure, particularly for you folks in nursing, is just ramping up. Getting worse. This is the little league. We're not in big leagues. Okay, I'll say it 1,000 times. The nursing instructors put a whole lot more pressure on you. Just wait till you get to that stuff. They're just vicious. And I understand why, and I'm in lockstep with it. I've lived that a lot. I'm only married to only two women in my lifetime. They both have lived that. Okay. So you understand. My first wife was this was at CCAC, which CCAC has a very good associate's degree program. I've taught many of those students over the years I've been teaching. I taught CCAC campuses for 17 years prior to just being here full time. Even when I was here full time, I was teaching here as an adjunct part time. She started there when there was one CCAC campus, the one on the north side, main campus, the Allegheny campus. She ended up getting an ulcer and going into hospital from nursing, having graduated. So it took her rather 2 years, because for a year or so she ended up having 2 abdominal surgeries, stressed out so much. So I understand the stress levels. Later, when she figured out how to be a good student and she was an auditory learner rather than a visual learner, she ended up graduating, getting her BSN in what's called the 2+2 program, when you start at Slippery Rock University here. She's one of the early classes, and she graduated with highest honors, went to the Nursing Honor Society, and all this happened when she was 31 years old. Some people late bloomers. Yeah, I mean, really, I mean, they learn how to study later than earlier. It's not that there's a certain amount of confidence, there's a certain amount of calm, there's a certain, you're stuck with me. I'm a professional student. My happy place is school. Okay. It's an old joke in the traditional Jewish families in New Jersey. Oh, what's Lewis doing? Oh, he's a sophomore. Oh, wonderful. You're always a student. You go from one school to another school to another school. What we do. Enough background. On to genetics. So everybody, that's the good news. Now back to work. Of the 3 chapters we do, I think the easiest to understand but requires a lot of memorization is the one we just did on germicides, disinfectants, antiseptic sterilization. The one that I emphasize the least is this one. Take that to the bank. 10 to 15 questions on the exam. So I'm not going to spend an inordinately large amount of time on it. There's 3 or 4 really important animations. The bulk of the exam is that one we just did and the one on viruses coming up. And viruses are very topical, and so really another really good look at diseases. Really going to start to talk about diseases and what they do a little bit more. So, how we go? Yeah, your genome is just your genetic makeup, and a gene is a subunit. And all living things have those. And for that matter, even the inanimate, the acellular forms like viruses have that. A genotype is your genetic makeup. A phenotype is something we can observe or measure. Hair color, eye color, freckles, dangling earlobes, all that stuff that typically genetics 101. If you haven't heard of these or you haven't done these, but you probably did. All that type of stuff that's there, the Punnett squares, things that people would do. We're not going to draw a segment of DNA or label anything. But we're not going to necessarily summarize bacterial replication, and we're not going to talk about leading or lagging strands. This genetic material overlaps what they do for the pre-med and bio majors. It's basically, did you cover that at all in AP1, where when they're assembling DNA, you have one strand right side up and one strand upside down, and there's directionality? Is this ringing a bell, anybody? One strand makes a smooth strand, the other one makes it segments, and you have to staple them together. No? Okay. Don't worry about it. So if I said Okazaki or Ozaki fragments, you would be looking at me and go, what are you talking about?
True.
Yeah. Japanese restaurant over at, sorry. Sorry, I'm having a sushi moment. Okay, heredity, buy it. Transmission, buy it. Traits are expressed, buy it. Structure and function, not so much. How it changes, mutations, coming up. So, all of our genetic material of an organism, most of it is in the chromosomes. Again, now we get to that difference between eukaryotic, and again, it's eukaryotic v prokaryotic. Plasmids, mitochondria and chloroplasts, where we have additional genetic material in eukaryotic cells as well as in the nucleus area. Okay, genome of the cells is about DNA. Viruses, RNA, and DNA, we'll deal with those in the viral chapter. So chromosome is just, and again, where's the distinction? And this is significant because of the sort of the idea of pairs that I talked about with dominant and recessive and things that may or may not be expressed. But the take-home point of eukaryotic chromosomes is it's DNA in pairs in the nucleus, membrane-bound organelle, and depending on, because we deal with mitosis and meiosis, which I know you've covered, typically we're going to be referring to diploid, less so about haploid. Understand now that haploid is what is in an egg or a sperm cell. It's a single strand of DNA, obviously, and fertilization is going to join with the mated strand from either the egg or the sperm, depending on who the donor is, who the recipient is, and form something called a zygote. Terms you should be familiar. You had to have heard them sometime before. That's going to be the genetic makeup, more or less, of that organism. Egg and sperm cells are haploid. And what's interesting about haploid cells is there's no pair, which means they're more susceptible to damage. Damage there would be expressed. Okay, that's the whole idea when you're getting an x-ray done. They tell us if you're pregnant, or they shield you if they're doing an x-ray study in the abdomen in a male or man or woman. They'll try to shield them as much as possible with lead shielding. In my office, we had lead aprons and all that type of stuff because you have to. Bacteria are haploid. They're a single strand of DNA, no paired strand. Number one. So inherently, they're more susceptible to mutation. Chromosomes in eukaryote are linear, obviously a bacteria, and also it's condensed. It's what we call supercoiled. So if you want, the question is, what's bacterial DNA like? It's a single circle that's supercoiled and haploid. Those three things, remember about it. So genes effectively code for proteins. There's a proportionately in our cells, 2 or 3% actually directs your body to make proteins, directs your cells to make specific. Bacteria is a larger percentage comparatively. We have a lot of what they call non-coding DNA. A lot of it is regulatory. That's when I talked about messenger RNA processing. When we make that big strand of messenger RNA, we basically chop out the stuff that's for coding purposes or management purposes and just staple the parts that are about making genes called exons together. So, and that also limits the number of mutations. So there's a lot of regulatory genetic material. Okay. All of those genes are the genotype that make them. So it's whatever is inside your genes, and the phenotype, again, how it's measured or observed. Something we can tell. That's what Mendel did. With all that Mendelean, the flowers and the pea plants, this color, did it wrinkle? Was it straight? Was it smooth? Whatever. Don't remember the studies. But I bet all of you had that somewhere in the past, right? He basically, a monk in Prague, without a computer, was able to develop concepts that we utilize today. Go figure. Just by doing it the old-fashioned way. Crossing them, basically planting them and letting pollinators pollinate them, etc., etc. So the difference here, it's double-stranded DNA, single circle here, different here because we have pairs. Okay, plasmids, small circles. Eukaryotic cells, we have them in mitochondria and chloroplast. Plants would have both. Our cells would have mitochondria. In really early eukaryotic cells, like protozoans and the early fungi developmentally, you might see some plasmids. That's why we can use that in DNA and basically using genetically modified organisms. We can even use yeasts. So viruses here, DNA or RNA never bind. Not going to get into the basis of this. Something you should have had. A lot of that is the directionality is based on something called 5', 3'. Don't panic. We're not doing it. I don't even understand it. I have a very brilliant colleague who teaches this in our cell and molecular genetic courses. I sit in on her lectures to learn about it. But it imparts this directionality I was talking about before. So we're not going to talk. We do always remember that A's and T's, G's and C's. So it's a simple alphabet, 4 letters for DNA, and there's a 5th letter for RNA. Remember, T's are always replaced by U's, uracil. Thymines, uracils, and cytosines are what are called, it's got a fancy name. Write it down. So the A's and the G's, and we get to some of this stuff, are called purines. Here a double ring, so you have two rings held together, and the C's and the T's and even the U are called pyrimidines. So they're just like a single unit
When do we see U's?
in RNA. DNA, so when you see, when we look at the genetic code a little bit, it will demonstrate, it'll be in terms of genetic code, when you look at it as U rather than T.
Okay.
So it's all about RNA, whether it's messenger RNA, transfer RNA, or ribosome RNA. It's all in that family. So the U stands for uracil. They're very, very similar. And down the road, when we start looking at antiviral agents, the earliest one was called 5-fluorouracil. And they would use that as what's called an anti-metabolite. You'd give that to somebody intravenously, and the fluorides, which are halogens, would effectively poison the RNA so it wouldn't work. So and hopefully the cancer cells would gobble it up before that's what a lot of chemotherapy was. And to some extent it still is. We call it passing poisons. You give somebody poisonous stuff and hope the ravenous cancer cells gobble it up faster than the good cells. Not scientific, is what you say. It's fairly medieval, but it's okay. That was the standard of care. That's an important thing. Standard of care is what you ought to be doing based on this, what your status of medicine is and wherever you reside and what everyone else is doing. You have to have an awfully good reason. This is important legally for many of you going into those kinds of fields. Why you have to have a good reason for not observing standard of care. It's the same thing for not using what's called the drug of choice. If you have a reason, it's fine. If you don't, you have to have a reason why you haven't used it or haven't done a particular procedure a particular way. This is stuff I don't do, but it gives you an idea. This is a pure, so A's and T's, G's and C's, you always have two of these units, okay, and one of these units. And that really was originally, if you should have gotten a little bit of this in AP1, this was the basis of the Watson and Crick model. What happened was, when I teach AP1, effectively, it was all about, we knew about the A's, the T's, the G's, and the C's. We just didn't know the arrangement. So we knew about the amounts that were in when they analyzed DNA. The ballpark here was never given, the lady who discovered it was never given credit. Her name was Rosalind Franklin. It's the tragedy of women in science and in medicine. Very few of them were recognized. It hasn't changed dramatically, but a little bit. And she did something unique. She basically took DNA and did something called X-ray diffraction. She took an X-ray of it, and she was trying to measure this so she could figure out how they would fit, because it was a consistent distance. And so what she did, since she couldn't get an X-ray that really worked, she took some DNA and froze it, I think in liquid nitrogen. So it would stop moving and she could calculate the distance. And so one day she came in at lunch and told Watson and Crick and another guy that she found out it was so many angstroms, whatever the unit of measurement. And all of a sudden the light bulb went off on their head. And they built a model, a Tinker Toy model. You can still see on any image on Google. They never credited her until almost, I think posthumous. She should have been part of that Nobel Prize. Absolutely, no question. It's another story. So, but we're not doing that. But the idea is, you see those little dashes in there? Hydrogen bonds. And remember, hydrogen bonds, they're a zipper. They open and close. Hydrogen bonds are very loose attachments. They're subtle magnetic attachments. It's almost like you taking a magnet that's stuck, pulling it apart. Under the right circumstance, it's easy to do, and it's easy to come back together. That's the critical piece about these double-stranded molecules. Okay. One's right side up, one's right side down. It doesn't matter where they're straight lines. We're not in geometry class. It's not two parallel lines and a transversal. All those angle, side, angle, or my favorite, angle, side, side. ASS. You didn't want me as a student. Told you, I was that kid who sat in the front row. Could you please be absent one day? No, it'll make your life too easy. I have my own chair in detention with my name on it. Goes around, comes around, baby. Okay, semi-conservative, all that means easy. This is stuff that I'm not going to ask you about. That doesn't mean shut off the brain. It means if you could get this on another test, like an NCLEX. Semi-conservative means when we make, whether it's bacteria or our cells, when they replicate, basically we use, we have an old strand and a new strand. Remember, the DNA unravels, okay? So you have two original strands. We make a new strand each one of those. So we always have an old strand and a new strand. If you think of it as like this. So I did this, I took this from a book and made a photocopy. If I ran this through the photocopy machine and then took that copy and ran it again, and then took that copy and ran it again, it fades. That's kind of what happens in cloning. You're just continually copying without any original material. It's a copy of a copy of a copy. And that's the problem with cloning. A lot of times as you get toward the end of the line, those cells or those clones are not as effective. They lose some degree of integrity. So that's why semi-conservative, don't worry about these. This is that one I was telling you about, proofreading the chain for mistakes. Okay. Most bacteria have DNA polymerase 1. DNA polymerase 3 is in eukaryotic cells. This one is going to basically repair mismatches. This one is going to proofread and repair mismatches. It's different. It's like you're doing a paper and you put spell check. Bacteria don't have spell check. I'm offended by spell check. My pet peeve with students, really getting it out there today, is spelling is deplorable, the sentence structure is deplorable, the grammar is deplorable. We had these teachers with the rulers. Yeah, don't do that. Stein, you're disrupting the class. That's what I live for. Stein, take a lap. Where should I take it? Take another lap. Thank you, I got two. It's my old gym teacher routine. Listen up, and I go this. What are you doing? You told me to listen up. Should I listen down or shall I listen straight ahead? The gym teacher and the janitor, they look the same. Gym teacher was a little more muscular. The gym teacher, khaki pants, soft pullover shirt, keys, 6,000 for all the lockers. Janitor had the same thing, except the janitor had one thing, didn't have one thing the gym teacher had, the whistle. You all had that gym teacher. Didn't you guys?
Yeah.
Even athletically, you still have them. The coaches out there. They're all the same. And if you don't do it right, take a lap. Where the F should I take it? We're not going to do that. We're not going to do that. See, we're not going to do elongation and termination and all that stuff, etc. That's why I don't love this PowerPoint, but it's in your text. But this is where we start to get to the important parts. Okay, this is the next section. But too far. Let's get to what they're talking about. Terms that you use a lot and need to remember: transcription and translation. Transcription is what DNA making any kind of RNA. It's an exact copy in RNA language, which means the U for the T. That's what transcription is. Before we had machines that could capture voices and you could listen to and type, secretaries did shorthand. They could, as fast as the boss or whoever's dictating the letter would speak, they could write it down and then basically transcribe it by typing it up. My mom did that professionally. She could take shorthand as fast as you could speak. She could type 120 words sustained copy. She was a professional secretary for the largest part of her life. Okay, that's transcription. So basically, it's an exact replica of the DNA in a slightly different language. Okay, translation is taking that RNA, typically messenger RNA, and applying it to the creation of proteins. Okay, so as you can see, and we'll get to what the viruses do. And there's a lot of different aspects of RNA. So we're not going to get into it, and we're not going to do all this stuff with this sort of chain of events with, and I don't know, again, whether you did it AAP or not, with ribosomal RNA and transfer RNA and codons and anticodons. DNA, the language is all three-letter words. RNA, the language is three-letter words. So, and even in transfer RNA, it's three-letter words. That's the terms. DNA, we talk about the three-letter combinations are triplets. In messenger RNA, they're called codons. The genetic code is based on those codons. And transfer RNA reverses them in RNA language. They're called anticodons. Again, I probably won't have questions about that, but down the road you might. And I really have to look at that as important. Okay. Like I said, to me, it's my success these days has to be your success. I'm probably not going for another job. Not doing this, the IE and T. Good. Okay, RNA, you got the uracil, fine, and the sugar changes, ribose to deoxyribose. You should know that. I'm not going to ask you. I'm not going to ask about any of that stuff. Any of that stuff. Okay. Again, the 70S for ADS we already covered. So there's not a lot here. This is important. Codons. So we take messenger RNA, and the beauty of this is a recurring theme for all of you who are in life science fields, whatever that field might be. Redundancy. We have so much I just was in my MCAT class. We're talking about kidney. You have two kidneys. One's redundant. You need 1/4 of one kidney to not have kidney failure. That's redundancy. You've got 7 times more kidney than you need. So much so, hey, you need one. Got you covered. Okay, that's redundancy. We see redundancy here. Okay. What they call, fancy term called wobble. I don't ask a question about that. Generally, it's so good that if you get, the code is so accurate that if you get the first 2 right, this would help you on your exams. If you get 2 out of the 3 right, you can have the 3rd one. No, we're not doing it. I'm not going to put this on. I'm not going to give you a strip. Other micro instructors might do this. To me, it's like I said, I minimize this particular chapter. I don't see it. I don't see it on anything. Yeah. But effectively, look at the first 2 letters. Again, it's the U rather than a T. A, G, C's, and U's. So all you have to do for most of these, pick the first 2, the last ones. The only thing that comes, so there's a lot of, generally there's between 2 and as many as 6 of these codes that apply. So almost all these amino acids have 2, and some have as many as 6. Here's punctuation. They're called stop codes. And there's a start code. And that's all, when we make the stuff, we always have a start code to start, and we always have a stop code. So it's just one giant sentence. It can be as long as the room, it can be this short. When we start to talk about what we want to talk about, the genetics, we have to understand that problems occur when either we have a mistake here in the lettering and we put the wrong one of these amino acids in, or we have a big mistake and all of a sudden we get a stop code where we shouldn't. And the protein doesn't get made at all. And they have very specific names. So when we start talking about mutation, and mutation is an error in DNA. When we get there, if for instance, if there's an error here, You have CC and all of a sudden the error is in U, C, A, and G. It makes no difference. You're still going to have proline. You don't have to know anything, the names of the amino acids for now. They might become relevant in some other way, but right now, not for our purposes. You're going to get proline, whether it's so that's called a point mutation, one letter, and in that case, a point mutation is known as a silent mutation, meaning even though there's a mutation, it didn't alter the protein. Happy dance. Like I said, your cells mutate all the time. Most of them are silent mutations because of the protection and the redundancy of this code. If, for instance, you were here, okay, and it was supposed to be an A, but it turned out to be a C, okay, you have a substitution. That is not silent because you're incorporating a different thing. So what we call that is a missense mutation. It may or may not have significant effects, but you definitely have a different amino acid in there. We're going to see this again in print. The classic example of that is sickle cell hemoglobin. Sickle cell hemoglobin versus normal hemoglobin has one misplaced amino acid. One genetic error can be a life-threatening condition. That's a missense mutation. Now, what happens if it was supposed to be cysteine and we all of a sudden had an error here, single error, and it was an A? We'd prematurely encounter a stop code. That's called a nonsense mutation because maybe that protein is supposed to be 30, 40, 100 amino acids long is now all of a sudden 6 and it doesn't do anything at all. Okay, that's a nonsense mutation. Those terms are important. Silent, missense, one alteration, nonsense, premature stop code. Nonsense mutations typically end up with big problems. And they're just showing you a little bit of this. We're not doing it. It is like an assembly line. You get this one strand and a whole bunch of ribosomes get onto it. And that's important to understand how rapidly proteins are made. For some of you, many of you, you're going to have the immune system. You'll probably look a little bit at this when you start talking about how the body generates antibodies so rapidly. No, and we're not going to do any of that stuff. So there's always a start codon. That codon is for an amino acid called methionine, but it's not the same methionine that's used later in the strand. Okay, that's of note. And so there's not a whole lot to show you anything was there. Here, pin loops involving carrying is transfer, but it's not a big deal for us. See if I have to know if I can answer the question. Not going to. This is. I used to have to teach this to nursing students at a community college. It's called the lac operon, and I'm not doing it. Be happy.
Thank you.
Okay, it's basically one of the ways that negative and positive feedback work. It actually is applicable to the lab. It has a lot to do with why when you start testing E. coli, why it loves lactose. It ferments lactose like crazy. And when you start to do selective and differential plates, the EMB plates. Did you do those yet? Okay, you're going to see that changes vivid colors. That's because it's a lactose fermenter. It has to do with that. We're not doing it. Okay. There's lac operon. Recombination. This is when we start to get into what these actually do. Recombination in our cells. Did you study meiosis? And in meiosis, we're creating haploid cells. Remember, we have like a tetrad. You duplicate the chromosomes, and here's the mom and here's the dad, and the ones that are next to each other kind of crisscross. Okay. There's a lot of terms for it: recombination, synapsis, crossing over, and it's where we include diversity. It's one of the ways diversity is assured. That happens in prophase 1. In metaphase 1, they line up along the plate randomly. They're not all mom in front, mom facing forward, or dad facing forward. It's completely random. Okay, so that's called the law of independent assortment. I'm not going to ask you about those, but bacteria do a different kind of recombination. It's what genetic engineering is based upon. Okay, and typically it's the donation of DNA from one to another. There's several different ways we're going to look at 4. They are really important. There will be questions on all of those. Okay. And I'll tell you the names now. We're going to look at each one individually. Transformation, transduction, conjugation, and transposition. Look at all of those. So, and what happens is if it recombines, in other words, if it's like someone's bringing it. I'm going to go over to St. Vincent de Paul down in Coriopolis with some stuff. Some stuff they don't take. Okay, most of the stuff they take. If they don't take it, it has no impact. If they take it, maybe they can use it, maybe they can resell it and make some money for charitable purposes. Okay, and so that's basically what recombination is. There's a lot of safeguards. So sometimes the DNA is accepted and sometimes it's not. And there's lots of things that move around. Resistance, Botox is moved around this way. Okay, the toxin part of it. So a lot of things. Any organism that contains genes that originated somewhere else is known as a recombinant. That's there. A quick review of plasmids. Small circular pieces of DNA. We'll look at origin of replication. Yes, when the bacteria's DNA replicates, the plasmid replicates. Typically by itself, independently. Sometimes they're joined. They are useful but non-essential, so that you don't have to have them survive. But typically things like resistance to antibiotics, developing certain poisons or mechanisms like that that might keep them alive longer is what they carry. Okay. And sometimes there's even little pieces of chromosome that move. We'll get to that as well. Those are the names. Conjugation. That's the one that involves that F pilus or F pilus. We'll look at a very good animation about that. Transformation has to do with free DNA. We'll get to that. And transduction is our introduction to viruses because the conduit, in science, we call that a vector. So everybody, if you're around here, you know one disease always is here, Lyme disease. Lyme disease is a bacteria in the bloodstream. It could be in the deer, it can be in a dog, it could be in you, it can be in a tick. The tick isn't bothered by it. The tick carries it from the deer or the dog in its bloodstream to you. When it bites you, you get the bacteria. It's called a vector. It's a way of moving it from one entity to another. In this case, it happens to be called a biological vector because it's in its bloodstream. When we look at a fly, that I have a lot of proof about this in my yard. When flies land on dog excrement, and then the flies fly around and land on food, that's also a vector because they're transferring bacteria from the excrement to the food. That's called a mechanical vector. It's not a bloodstream. Fine point, but easy, not too hard to remember. But it's a biological vector for Lyme disease. That typically is a question down the road. So we take a look at this. And we'll look at the F pilus. I don't have a lot of time. I don't want to start a whole lot of new stuff. Yeah, I don't want to go through all those yet. Let me flip over to this other one for a little more information. Because this is the introductory stuff. It's going to take probably all 3 periods of this week to do this chapter, I would estimate. This is Nestor. So again, you'll see that it'll look like this if you conjure it up. This is another lady in science who basically observed the corn that, it's a pejorative term again. We used to call it Indian corn. Is that ringing a bell? Native American corn these days, of course. But you have the corn that's on, people put in their doors, Halloween time in the fall. Okay. Though she observed these patterns repetitively, and the only way she could figure out, because she tried to study how they could be inherited and where they would appear, was that DNA segments had to literally move in and out of genes from one place to another for it to have some of these identical but inconsistent patterns. Okay. And she published her results in 1950. She's a lady. So people thought DNA is stable. It's not. The idea became accepted by the 70s, and while she was still alive, thankfully, she was awarded Nobel Prize in 1983. And so they're called transposons for transposition. We'll see an animation about that. That's a relatively unique way that genetic things can move. It played a very significant role in bacterial resistance, which we will get to. We get to that more in the antibiotic section. So this is a lot of different things. This is a very good slide that shows you all the DNA in that sort of yellowish color, if you're seeing it there, inside a bacteria that effectively has died and the DNA has exploded out. That is so supercoiled it would wrap around that E. coli about 1,000 times. So imagine how coiled up it is if you can wrap that around that microscopic entity 1,000 times. So we're going to look both at mutations and we're going to look at movement. When they say horizontal gene transfer, it's not the way we transfer genes. We transfer genes vertically. We transfer it from parents to offspring. The only time we get any kind of horizontal transfer of important material might be a nursing mother, as much as anything else, unless we're doing it on a laboratory basis, taking antibodies from one person and inject them into somebody else. That would be a horizontal transfer to some extent, but it's not a genetic transfer, certainly. So, but again, bacteria have horizontal and what we typically call lateral, side-to-side transfer. And this again is the mechanism that a single MRSA, a mutated Staphylococcus aureus cell, can make this whole room become MRSA overnight. Through all non-MRSA. It is that rapid and that significant a phenomenon. The graph doesn't help me. So again, genotype and phenotype. Down the road, these are things that I do not know if they use them in the lab, but it's terms that you're going to become familiar with a little bit. There's auxotrophs and prototrophs. So you're going to see one of the things that we learn about, it's a good stopping point, here is called the Ames test. Anything that the Food and Drug Administration has called it. Food Administration, Drug Administration is the Ames test. They take a salmonella, that's a very nasty bacteria, called auxotroph. That means it requires histidine. You'll see the script. That's a mutation. We use that bacteria when we're testing an agent that we could use as a medicine or something we could use part of us by this Ames test. We mix it with living cells and see if it can make, frankly, if it can cause cancer in a laboratory. Okay, it does so by creating what's called a back mutation, a reverse mutation. If it's able to they can turn it back to normal salmonella. We know it's so strong of a mutagen, mutational agent, that we don't want to have it in stuff. So we're going to look at that. That's where that becomes significant, that language. Let's leave it here, and we'll pick up on the different genetic mechanisms of transfer very soon. Take your things, everybody, please. Just take one. If the people who weren't here, they'll get them on their own time, I'm sure.